How the pregnancy test is work. The earliest known pregnancy tests date back to 1350 BC in ancient Egypt. According to Egyptians, all you have to do is urinate on wheat and barley seeds and wait. If others sprout, congratulations, you are pregnant. And if wheat sprout faster, it's a girl. But if barley, it's a boy. In 1963, a small study reproduced this test and found that it predicted pregnancy with a respectable 70% accuracy, though it couldn't reliably tell the sex of the baby. Scientists hypothesized that the test worked because the pregnant woman's urine contains more estrogen, which can promote disease growth. Now, it's easy to take these ancient methods for granted, because modern pregnancy tests give highly accurate results within minutes. So, how do they work? Over-the-counter pregnancy tests are all designed to detect one thing: a hormone called HCG. HCG is produced in the earliest stages of the pregnancy and starts a game of telephone that tells the body not to shed the inner lining of the uterus this month. As the pregnancy progresses, HCG supports the formation of the placenta, which transports nutrients from mother to fetus. The test starts when urine is applied to the exposed end of the strip. As the fluid travels up to the absorbed fibers, it will cross the three separate zones, each with an important test. When the wave is the first zone, the reaction zone, Y-shaped protein called antibodies will grab onto any HCG. Attached to these antibodies is a handy enzyme with the ability to turn on dye molecules, which will be crucial later down the road. Then the urine picks up all the AB1 enzymes and carries them to the test zone, which is where the results show up. Secreted to the zone are more Y-shaped antibodies that will also stick to HCG on one of these 5 binding sites. Scientists call this type of test a sandwich assay. If HCG is present, it gets sandwiches between the AB1 and enzyme and the AB2 and stick to the test zone, allowing the attached dye activating enzyme to do this job and creates a visual pattern. If there's no HCG, the wave of urine and enzyme just passes on by. Finally, there's one last stop to make: the control zone. As in any good experiment, this step confirmed that the test is working properly. Where the AB1 enzymes never saw HCG, or they're extra because the zone 1 is overstocked with them. All the unbound AB1 enzymes picked up the zone 1 should end up here and activate more dye. So if no pattern appears, that indicates that the test was faulty. These tests are pretty reliable, but they are not fail-proof. For instance, false negatives can occur if concentration of HCG aren't high enough for detection. After implantation, HCG level doubles every 2 to 3 days, so it may just too early to tell. And beverages can dilute the urine sample, which is why the doctor recommended taking the test first thing in the morning. On the other hand, false positives can come from other sources of HCG, like IVF injection, ectopic pregnancy, or certain cancers such as urinary cancer or testicular cancer, making it possible for one of these tests to tell the man he's pregnant. The best way for a woman to find out for sure is at the doctor's office. The doctors are also looking for HCG, but with the tests that are more sensitive and quantitative, which means they can determine the exact level of HCG in your blood. A few minutes can feel like forever when you're waiting on the result of a pregnancy test. But in that brief time, you're witnessing the power of the scientific methods that one little stick lets you ask the question, perform a controlled experiment, and analyze the result to check your original hypothesis. And the best part is, you won't even have to wait until the next harvest. One more as the original speed. The earliest known pregnancy test date back to the 1350 BC in ancient Egypt. According to Egyptian, all you have to do is urinate on the weak and barely see and wait. If others sport, congratulations, you're pregnant. And with sport, the factory circle, and barely, it's a boy. In 1963, a smart study reproduced this test and found it predicts pregnancy with a respectable 70% accuracy, though it could reliably tell the sex of a baby. Scientists hypothesized that tests worked because the pregnant woman's urine contains more estrogen, which promotes seed growth. Now, it's easy to take these ancient methods for granted because modern pregnancy tests give the highly accurate result within minutes. So, how do they work? Over-the-counter pregnancy tests are all designed to detect one thing, a hormone called HCG. HCG is produced in the earliest stage of a pregnancy and the start to game or telephone that tells the body not to shade the inner lining of the uterus that month. As the pregnancy progresses, HCG supports the formation of the placenta, which transfers nutrients from mother to fetus. The test starts when urine applied to the exposed end of the strip. As the fluid travels up to the absorbed fibers, it will cross the three separate zones, each with an important task. When we hit the first zone, the reaction zone, one-shaped proteins called antibodies will grab onto any HCG. A test of these antibodies is a handy enzyme will be ability to turn on diamide molecules, which will be crucial later down the road. Then the urine picks up all the AB1 enzymes and carries them to test zone, which is where the results show up. Secreted stones are more wide-shaped antibodies and they also stick to HCG on one of the 5 binding sites. Scientists call this type of test a sandwich assay. If HCG is pregnant, it's to get sandwiches between the AB1 enzyme and the AB2 and stick to the test zone. Allowing the attacks the dye-activating enzyme to do this job and create a visual pattern. If there's no HCG, the wave of urine and enzyme just pass on by. Finally, that's one last stop to make the control zone. As in any good experiments, the stop confirms that the test is working properly. Whether the AB1 enzymes never saw HCG or they extra because zone 1 is overstocked with them. All the combined AB1 and enzyme pick up the zone 1 should end up here and activate more dye. So if no pattern appears, that indicates the test will faulty. These tests are pretty reliable, but they're not verifiable. For instance, false negative can occur if concentration of HCG are high enough for detection. After implantation, HCG's level doubles every 2 to 3 days, so it may just too early to tell. And babies can dilute urine sample, which is why doctors recommend take the first thing in the morning. On the other hand, false positive can come from other sources of HCG, like IVF injection, atomic pregnancy, or certain cancers such as urinary cancer or testicular cancer, making it possible for one of these tests to tell the man is pregnant. The best way for women to find out is sure is at the doctor's office. The doctors are also looking for HCG, but with the tests that are more sensitive and quantitative, which means they can determine the exact level of HCG in your blood. A few minutes can feel like forever when you're waiting on the results or a pregnancy test. But in that brief time, you'll witness the power of the scientific methods that one little stick lets you ask a question, perform controlled experiments, and analyze the results to check your original hypothesis. And the best part is, you won't have to wait until the next harvest.